going to talk about lysosomes. So lysosomes are membrane-bound vesicles which contain a variety of digestive enzymes. So in fact, while we were discussing about the significance of Golgi apparatus, so they are also vesicles, they are also vessels which are surrounded by one membrane, single membrane bound, and they help to store the digestive enzymes. So that is their most important job, because these digestive enzymes are extremely important. A lot of digestive inappropriate activity might happen inside the cell, because whenever we eat, the food material is passed on, it is digested and then it is passed on to various parts of the body. So what do we mean by various parts of the body? End of the day, the food material actually goes to each and every cell of our body. So the cell has to handle that food material. So to take care of any appropriate digestive activity, we have these lysosomes. So let us see where are they located. So the lysosomes are located here. So these are small vesicles. So here you can see this structure which you see here is the lysosome. So what are these digestive enzymes which we are talking about? What kind of digestive enzymes are present inside lysosomes? So these enzymes could be nucleases which digest nucleic acids like RNA or DNA. It could be proteases which digest the proteins. It could be lipases which digest lipids. It could be glycosides. So glycosides, they digest polysaccharides, that is glucose. Glucose is a monosaccharide, so the complex sugars, the polysaccharides or the disaccharides. So these kind of enzymes are contained inside the lysosome. Let us see where exactly is lysosome present in animal cell. So lysosomes are present quite near to the mitochondria. So this is the lysosome. In plant cells, the presence of lysosome is disputed. So some say that lysosomes are present in plant cells, some say that they are not present in plant cells. Let us quickly look at the significance. So the lysosomes digest waste materials or worn out organelles. Now whatever waste materials are present inside the cell or the organelles which have been damaged, so it helps the cell to get rid of all these waste materials because it contains the digestive enzymes. So these enzymes can digest all unnecessary material, and that means it can clean the cell. Defense against bacterial and viral infection, because these bacteria or virus which can cause infection or which can damage the cell, so they can be destroyed by the action of these digestive enzymes. Waste disposal system of the cell. So as I had mentioned before also, it helps in getting rid of the waste. During cell damage, lysosomes burst and the enzymes digest their own cell. Now what happens here is, as I said, inside the lysosomes are present various digestive enzymes like proteases, lipases, nucleases, phosphatases, etc. Now without a constant input of energy, these enzymes, they start digesting the membrane of the lysosome from inside. So let us suppose if this is your lysosome and inside the lysosome you have these enzymes. Now when there is no input of energy, so there is no energy, so what will these enzymes do? They will start digesting these walls. What I'm trying to explain you is, without a constant input of energy, how the cell gets damaged, and that is why cell needs energy. So when there is no input of energy, these enzymes will start eating up, or it will start digesting the membrane of the lysosome, because the membranes are mostly made up of lipids or proteins, and the enzymes which are present inside, they are capable of digesting those components as well. So it will start digesting the membrane. Now when the membrane completely disintegrates, what happens? These enzymes which were till now present inside the lysosome, these enzymes now are released out into the cell. Now when they are released open in the cell, so these enzymes can very easily start eating up everything which makes up the cell. Because the cell, various cell organelles, the cell membrane, everything is made up of one of these components, nucleic acids, proteins, lipids. So these enzymes will start digesting all of them, and finally the cell will be destroyed. And that is why lysosomes are called suicidal bags of the cell, because the enzymes present inside the cell can destroy that cell only. So that is why they are suicidal bags. Suicide means when somebody kills himself or herself, that is called a suicide. So here also the cell gets destroyed because of the enzymes which were present inside that cell only, because lysosomes are part of the cell and these enzymes are present inside the lysosomes. So lysosomes have advantages because it helps to get rid of the waste material. So it is like the waste disposal system for the cell. But at the same time, during lack of energy, due to lack of energy, these lysosomes can become the suicidal bags for the cell. So that is why they are termed as suicidal bags of cell. About the next cell organelle, that is Golgi apparatus. So the name is quite a little weird. That's not because it is related to some of its function, but it is named after the scientist who described it for the first time. So it was named after Camillo Golgi, who first described it. So what are these? So these are again network of membrane bound vesicles arranged parallel to each other in stacks. So look-wise, Golgi apparatus also looks like a similar network like that of endoplasmic reticulum. So there also you saw a network of tubules. Here also you will see network of vessels. But here each vessel, they are arranged parallel to each other. So something like this, you have one vessel, two vessel, three vessel, four vessels. So they are like arranged parallel to each other, like in stacks, one after another. So this stack in which the vesicles are arranged, they are called cisterns. So this is the term which is given to the stack of the vesicles which are arranged parallelly. And such network together form the Golgi apparatus. So these are mostly seen in cells that are actively involved in secretion of chemical products. So secretion is one of the important role of Golgi apparatus. So we will look at the functions of Golgi apparatus. But before that, let us see where are they located. So they are also located near nucleus in animal cells. And in plant cells, they are freely distributed, and in plant cells, they are often called as dictyosomes. So the name of this particular cell organelle is different in plant cells and in animal cells. In animal cells, it is called Golgi apparatus. In plant cells, it is called dictyosomes, or it is also called as Golgi body. So these are different names for the Golgi apparatus. So let us see where is it located and how it looks. So this is how it looks like. So you see, these are the various vesicles. So each of these is a vesicle. 1, 2, 3, 4, 5, 6. So they are like different vessels and they are all arranged parallel to each other. So they are arranged in stacks and each of these stacks are called cisterns. So you see, this is one cistern where you have so many stacks arranged like this. Similarly, this is another cistern. So a lot of cisterns together form the entire network. So you see, these are all cisterns and each of these are like stacks of vessels. Now the number of cisterns in a Golgi apparatus can vary. There is no specific fixed number of cisterns that has to be present in a Golgi apparatus. So here you see cis phase and trans phase. So these are like the two sides of the Golgi apparatus are named as cis phase and trans phase. So which is a cis phase, which is a trans phase, I think that is not really required at this level. So you just know this, that this is how the Golgi apparatus will look like. It will consist of a lot of vesicles that are arranged parallel to each other. One side is going to be cis phase, the other side is going to be the trans phase, and that is how they are going to connect to each other. Now where are they located in the animal cells? Now as I said, they are also located somewhere near the nucleus. So let us try to find them. So here you have the Golgi apparatus or the Golgi vesicles. Here you see the green colored structure. So just outside the endoplasmic reticulum, you have this structure. So these are the vesicles which all together form this network of Golgi apparatus. Similarly, they are also present in plant cells. So I'm sorry, here by mistake it is written as animal. So it is plant cells. So where do we find them? In the plant cells. So let us look for the Golgi body or the Golgi apparatus or the dictyosomes. Now as I mentioned before that they are freely distributed in case of plant cells. So here you can see So this is your Golgi apparatus. Other than this also you have these vesicles scattered here and there. So that's how they are located, and that is why they are called dictyosomes. So we have given a different name because of their different arrangement in case of plant cells. Now what is their significance? What role do they play? So these Golgi apparatus, they help in transport of material. So the materials which are manufactured in endoplasmic reticulum is dispatched to various parts inside or outside the cell through Golgi apparatus. So Golgi apparatus is like the courier service. So we prepare something at our home and then we want to parcel the same thing to our friend who lives at some other place. So what do? How do we parcel it? So for that, we either need a courier service or we need a delivery boy who can go and just make it reach there. So in a very similar way, here we saw that in endoplasmic reticulum, a lot of substances get manufactured like proteins, lipids, they all get manufactured in the endoplasmic reticulum. Now once these substances are getting manufactured, they are needed by different parts of the cell. So they need to reach different parts. So these substances need to be packaged and then sent to different parts. So this packaging is taken care by the vesicles. So they are packed into these vesicles and then dispatched to various parts inside the cell as well as outside the cell. So the packaging and transport is taken care by the Golgi apparatus. So packaging is one function of the Golgi apparatus. It also helps in storage of various cellular secretions like enzymes, proteins, cellulose, etc. So the secretory proteins and lipids, they are packed into the secretory vesicles. Now these vesicles also are of various types. So there are something called secretory vesicles which will store only the secretory proteins and lipids. So that means we have different types of vesicles for storage of different types of secretions. Now pectin, which makes up the cell wall. So pectin is one of the important component of cell wall. Cell wall is present only in plant cells. So pectin is also secreted by Golgi apparatus. So pectin, which is an important component of the cell wall which is present in plant cell. So this pectin is secreted by Golgi apparatus. So that is also another important thing. So we can say that packaging was first role and then secretion could be the secondary role of Golgi apparatus. It also helps in synthesis of lysosomes. Now you might ask, what are lysosomes? These lysosomes act as storage vesicles for digestive enzymes. So they act as storage vesicles for digestive enzymes. Now digestive enzymes are extremely important, right? Because the process of digestion is very, very important. So these are like small single membrane bound spherical shaped vesicles. They will help in storing the digestive enzymes. Now these lysosomes can protect the rest of the cell from any kind of inappropriate digestive activity, because whenever there is something inappropriate happening with the digestion, then these lysosomes can anytime release the digestive enzymes which are present within it. And these lysosomes are synthesized or manufactured by Golgi apparatus. So that is also one of the very important function of Golgi apparatus. So we can say that packaging, secretion and lysosome synthesis are the main functions of Golgi apparatus. Now that we have this picture of Golgi apparatus, let us quickly see how exactly it helps in packaging and transport. Now here you can see an incoming transport vesicle. So what happens is, whatever material needs to be packaged, that is sent from the endoplasmic reticulum. So endoplasmic reticulum is actually manufacturing the proteins and the lipids. So all those materials will come through the incoming transport vesicle and then it will enter inside the Golgi apparatus through the cis phase. Then from the cis phase, it will reach the cisternae. What will happen in the cisternae? These cisternae, modification of proteins will take place. Now, before it is actually being transported to rest of the body, if any modification is needed, all those modifications will be done in the cisternae. And finally, it is sent to the trans phase, and from the trans phase, it is sent out through the outgoing transport vesicles. So here you have the outgoing transport vesicle. So where will this outgoing transport vesicles take it? It will take it to the cytoplasm, and from cytoplasm it can be taken out through the plasma membrane. So from the plasma membrane, it can even be taken outside of the cell. So that is why it was told that it helps to transport substances not only to various parts inside the cell, because inside the cell it can reach through cytoplasm. In fact, it also helps to send it outside the cell. So once it goes out through the plasma membrane, then it can reach outside the cell. So that's how Golgi apparatus helps in packaging and transport of substances which were manufactured in endoplasmic reticulum. That is why the Golgi apparatus is located quite close to the endoplasmic reticulum. Important organelle is mitochondria, and this mitochondria, they are called the powerhouse of the cell. So let us talk about it. So these are rod-shaped organelles bounded by two membranes. So here you have one outer membrane and one inner membrane. So you see here, this is the outer membrane and this one is the inner membrane. So this is outer membrane and this one is inner membrane. And in between these two membranes, the space which is there, that is called the peripheral space. So this entire structure which you see in this on the screen is the mitochondria. Now, why is it named mitochondria? The word mito, it means thread. And the term chondria, what does it mean? It means granule. So it is something like some thread-like structures with granules over it. So that is why it is mitochondria. So we will get to know this more when we actually look at its structure. So you see the granules here are in the form of ribosomes, which are present in the form of granules over the mitochondria. And what are the threads? So these structures which you see on the mitochondria, they are like a mixture of threads or combination of a lot of threads. So that's why it is named mitochondria. So the number of mitochondria which can be present in one cell is variable. So one cell can have one mitochondria, one cell can have multiple mitochondria. So the number can actually vary. If you talk about its shape, it is mostly cylindrical in shape. So mostly cylindrical in shape. Sometimes it is in the shape of a sausage. So mostly either like this or like this. So something of this sort. So generally mitochondria is uniformly distributed in the cytoplasm. So it is not that if this is your cell, so mitochondria is more or less uniformly distributed. It is not that in some area there are a lot of mitochondria and in the remaining areas there are no mitochondria. So that is not the case. And it is always surrounded by a double membrane, as you can see, outer membrane and inner membrane. And chemically, if you look at the composition of each of these membranes, they are similar to the plasma membrane. That is, these membranes are also made up of lipids and proteins. In fact, if you look at it, most of the membranes are made up of lipids and proteins. So now let us see where exactly they are located in animal cell. So in animal cell, this is where you have mitochondria. So this yellow structure, you see the entire yellow structure with a green structure inside. So this entire thing is the mitochondria. So here in this particular cell, you can see two mitochondria. This is another mitochondria that you can see here. So that's why I told that there can be multiple mitochondria present inside a cell. In plant cells also mitochondria is present. So this is mitochondria. So this is one mitochondria, this is another mitochondria. Now what is the significance of mitochondria? So these mitochondria are called the powerhouse of the cell. Now why are they called powerhouse? Because this particular structure generates energy, and that's how the cell receives energy. So just now in the previous topic when we were talking about lysosomes, we were talking that if there is lack of energy, if there is no input of energy, in that case the cell gets destroyed. So the cell needs constant input of energy. So from where will this energy get generated? So somebody within the cell needs to generate the energy. So that energy is generated by mitochondria. So let us see how important are they. So as I said, they are the powerhouse of the cell. Why? Because they release energy in the form of adenosine triphosphate molecules. Now we all know what is ATP. Even if you don't know, so just know this, that ATP is nothing but the energy currency of our body. Like, what do we mean by currency? For example, you want to buy something. So how do you buy your stuff? You have to give your currency note. Now this currency can be rupee, this currency can be dollar, it can be pound. So the currency can be different, but you need some currency to exchange stuffs. Similarly here, how do we measure energy? That okay, how much energy is being produced? Now, for measuring energy, we have a unit called joule. But when we talk about production of energy inside the cell, we talk in terms of a energy currency, and that energy currency is ATP molecules. So we talk in terms of number of ATP molecules which are being produced, or the number of ATP molecules which gets utilized in a particular process. So ATP is a molecule that is adenosine triphosphate. So 3 phosphate groups attached to the adenosine molecule forms adenosine triphosphate. So you will learn about the structure and etc. chemical composition of ATP in your higher classes. So what mitochondria does, it actually converts glucose. So this is what happens during the process of respiration. So glucose is oxidized to produce carbon dioxide, water and energy. And this energy is nothing but around 38 ATP molecules. So that is what happens during respiration. And this generation of ATP molecules takes place in mitochondria. That is why mitochondria is called powerhouse of the cell, because it supplies energy. So it gives power to the cell. So it is the powerhouse. It can synthesize its own proteins as they have their own DNA and ribosome. Now for synthesizing proteins, one important thing is ribosome, because they are the site of protein synthesis. And for protein synthesis, we also need DNA. So from DNA, RNA will be produced, from RNA, proteins will be produced. And both of these are present inside mitochondria. Therefore, mitochondria can synthesize their own proteins. They are not dependent on the proteins which are coming from other organelles. Like we saw that endoplasmic reticulum, they also synthesize proteins and lipids. And then they are packaged and sent to other parts by Golgi bodies. So mitochondria is not dependent on anybody else because it can synthesize and produce its own proteins. So that is why mitochondria is called the powerhouse of the cell. So here you can see there are some specific particles called ATP synthetase particles which are present in the mitochondria, and they help in the process of ATP synthesis.